and thank you so much for rejoining us. We are now on module 4, and we're talking about the fascinating world of nucleotide nutrition. I'm joined by Rachel Hoyle, and we're so proud to have Rachel with us and her amazing company. They are CMA approved suppliers, which is essentially a massive badge of honor that only goes to companies that really truly are going well far and above the call of duty when it comes to the level of services and expertise and excellence that they provide within the complementary medical and integrative medical sectors. So Rachel, thank you so much for joining us again.
Thank you, Janie.
It's such a pleasure to have you. In this module, everybody, we're talking about the important roles of nucleotides and the evidence. So Rachel, could you explain a little bit about what the evidence base actually is at the moment for nucleotide supplementation?
Oh, it would be a pleasure, yes. Well, the evidence base actually does go back quite a long time. I mean, it's been a really neglected area of nutrition, but there are some areas where it's been less neglected. Indeed, I mean, my involvement with nucleotides goes back to the late 80s in my first job, which is where I first struck up a collaboration with the Swiss experts in nucleotides, and have forged that collaboration ever since then till now. I've been working with them to provide products, nutritional products based on nucleotides. So, but the work that we all saw that had been done with nucleotides was with infant formula. A lot of work had been done by these companies looking at the content of mother's breast milk, and what they found was that in breast milk were high content of nucleotides, particularly the pyrimidine nucleotide. So they did a lot of clinical trial work looking to see what impact adding in purified nucleotides into infant formula had compared to those infants that were luckily breastfed. What they found was that by including the nucleotides, the results in terms of digestive health and the development of the immune system of the babies was much better than the infant formula that didn't have those nutrients in there. So there's a lot of work that's been done on the importance of nucleotides for infants, and that's why infant formula has the nucleotides, the 5 nucleotides supplemented into the formula. So that's the first area, which gave a big clue. And then what we did actually start working with animals, particularly stressed farmed animals, who were reliant on antibiotic growth promoters to support their health and growth. And there was a big drive to start to find natural replacers of these types of what we call them, drugs, really. Drugs that we all would rather get rid of because there was implications for resistance, antibiotic resistance in humans. So it was a big drive to get rid of them. So we got involved with a lot of research with farm animals, poultry, pigs, salmon. And what was found was that where the nucleotides was supplemented into the diet of these animals, they were able to show a much better functioning gut, less lesions, far superior absorption, and subsequently mortalities, and their health was improved. So whether you're, a fan of farmed animals or not, their lives were improved by including nutrients such as nucleotides, not just nucleotides, there's a whole range of nutrients that they need to support their health. But we were involved in research to try and reduce the number of times that antibiotics had to be used with farm animals. So we learned a lot from that work. So because in the end we are all animals, And so we had a big clue that if you supplemented with nucleotides, you could improve the health of the gut, the repair and maintenance and absorption of nutrients, as well as the response of the immune system as well, because these areas are areas where these cells absolutely require dietary sourced nucleotides. So I suppose that's where the initial research was, and then we went on and we managed to get grants to take the formula that was used of nucleotides with the animals. We developed that to suit more the deficiencies that humans were facing in their foods, and we did some clinical trial work to try and prove, if you like, that supplementing with particular types of people in certain situations, they would yield a benefit from taking them.
Yes. And Rachel, can I ask you where in the body, just to recap on this, because we touched on it in an earlier module, where in the body, particularly the human body, as we're speaking about that predominantly at the moment, would supplementary nucleotides have the greatest effect, do you think?
Yeah, well, it's generally where there's the highest demand for new cells. Generally also short-lived cells, so gut cells only last sort of about 4 days. Immune cells are constantly being needed, especially if there's an infection going on around. Fast replicating cells, so high demand, short-lived, and fast replication. So those are the ones that tend to need to either top up their need for nucleotides, or they can't make nucleotides at all, so they need to get them from the diet anyway. So bifidobacteria, for instance, would need to get nucleotides from the diet.
Right. Okay. Yeah. So what would you say is the first impact of supplementation? Presumably, would that be the intestinal flora then? Because that's, I guess, where it's going, isn't it, first?
It is, yeah. So there's the intestinal flora. There's the cells that form the lining of the gut. They need the nucleotides from the diet. So those pools at the base of the villi, in the crypt, the nucleotide pools, so they're replenished from nucleotides coming in from the digestive process, and they form the new cells of the gut lining, and also the nucleotides get involved in the repair processes in the gut. And then you've got the white blood cells, those ones particularly in the bone marrow. They all need to have dietary source nucleotides, because they're you know, if you have an infection, you have a vast number of new cells that are required, and so they've got to build DNA and RNA, and that takes, as we know, 3 billion. DNA has over 3 billion nucleotides, so they always top up their requirement from the diet. And then as part of the bone marrow, there's the red blood cells as well.
Yes, of course.
So those would be the 4 main areas. Yeah.
Yes. Now, I know that obviously you were saying, just rewinding to what you were saying about how you kind of came into this work, and it was also through looking at the impact of nucleotide supplementation on farmed animals. And I remember you saying that they had 25% higher villi, and after just 3 weeks of supplementation, fewer lesions and greater absorption of nutrients. And of course, obviously, that then leads to reduction in the requirement for antibiotic growth promoters and so on. But what about us humans? Obviously, you can't really ethically do too much sort of histological analysis and take cells out and look at them under a microscope. So how have you managed to conduct research in human subjects?
Yeah, well, what we've done is we did a clinical trial with sufferers of IBS, and they were diarrhea-led IBS participants. and did a symptom scoring trial basically with them. And so it was quite classically designed, that there were 39 participants, all with diarrhea-led IBS, and they were split into two groups and either had the nucleotide or the placebo. Then after the 6 weeks of taking those every day and reporting on 7 main symptoms, they then had a rest period, and then they crossed over. So everybody had a chance to be a placebo and take a nucleotide supplement. And what they found, the researchers found with that in this published work, which I can provide everybody, is that the symptoms that were reported that had been significantly affected, statistically significantly been affected, were abdominal pain, urgency, diarrhea, and feelings of incomplete evacuation. And what the researcher surmised from that was that those were the symptoms that are often affected because of poor motility. So the movement through the gut is, if that is impaired, then those are the symptoms that tend to be heightened. So she surmised from the work that had been done with the animals that if you can actually improve the repair and the maintenance and the health of the gut, you actually get an improved motility, movement. And that's what had been shown by the improvement in those types of symptoms. So though we haven't done with humans any direct sort of interaction in the gut to actually sort of measure things like lesions and leaky gut and that, I mean, hopefully we will be able to in the future. Need to try and find some more money to do that, but we decided to select participants who had a gut disturbance and see whether we can actually improve their lot, if you like, by taking the nucleotides.
That would make such a difference to the quality of life. And understand also that you've had some support from health practitioners, but I'm sure you would probably quite like health practitioners to get onto this even further for the future. But as I understand it, you've been working with health practitioners now for over 10 years, and you've built up a sort of portfolio of case studies, a number of different gut conditions. Can you tell us a little bit about that?
Yes, I've got some health practitioners who have been working with me for quite a number of years, and I always ask them if they could provide me with some case studies, because it's one of the first things that often people who are new to the science, health practitioners who know what nucleotides are but never thought about using them in their protocols, they really want to see case studies, and so I try and get as many as I can, covering a variety of different conditions. And so things like obviously IBS, where we've got the clinical trial, we've also got case studies there, because IBS tends to have different reasons. People getting IBS don't necessarily get it for the same reason. It might come from a food poisoning situation, or it could be just stress overload over a number of years. All sorts of different reasons why people might be lumped into that IBS sort of pool. And then ulcerative colitis and celiac disease. So people who are celiacs will have damaged guts, and then they come, that's established, and they come off, they take gluten out of their diet, they've still got damage in their gut and they need help. And sometimes, you can't always eliminate gluten. Something comes into your diet that's got gluten, and then you've got damage again. So, you know, there's all sorts of areas where we've managed to get case studies from health practitioners, and it really helps people understand where it might work for their, if you like, clients and patients.
Yes, absolutely. And tell me a little bit more about immunity and metabolic response. Obviously, we know that high demand, short-lived, fast replicating cells require massive amounts of nucleotides. But how does that also, can we extrapolate and assume that that also applies to the cells of the immune response?
Yeah, definitely. I mean,
very topical right now.
Yeah, very helpful right now, definitely. I mean, the body's natural reaction when a virus or rogue bacteria enters the body is to produce a variety of different cells in order to vanquish it, and then also produce memory type immune cells, so that if it comes around again, then it works even quicker. So all of those cells, I mean, and there's so many, a lot of them are produced in the bone marrow, and as we talked about before, the bone marrow needs nucleotides from the diet, and so if they're in short supply, there is a good chance that the immune response will be made more sluggish, because it's still waiting for all the different nucleotides to come along to pair up to form the new DNA and complete the cell renewal and rapid expansion that the immune system needs to vanquish these different infections. So yeah, if there's a shortage of nucleotides, there's a good chance that your efficient response to infection will be impaired.
Rachel, in that case, can I just ask you, if one was faced with a pathogen, and you know, we of course realize that we can create an upregulation of appropriate immune response, because obviously what we don't want to do is to take somebody, let's say, who's in danger of sort of descending into a cytokine storm. Obviously, at that point, we've got to modulate the immune response and calm that down. And that's working really well with things like IL-6 suppressors and anti-TNF meds and so on. So, you know, so that's actually working very well from a conventional perspective. But if somebody was faced with getting something like an early stage ailment, a cold, a flu, or dare I say anything that may be circulating at the moment without naming names, would it be then prudent in that case? And I appreciate you can't say because we don't have the research, but in your observations, would it be prudent in an early stage infection to actually supplement with extra nucleotides, with extra nucleotides?
It would. We have actually got a clinical trial that was done up at Edinburgh, University in Edinburgh, where it was actually good. It was at a university, because you've got a lot of young people together. They tend to, you know, if there's a cold going around or flu going around, a lot of people tend to get it. So what you want to do in a trial is try and get enough of the same bug to get a, you know, to be able to research it properly. So what the researchers did was they put a note out to them all to say, if you've got a cold or flu, come down to the laboratory, we'll take a saliva test and make sure you've actually got a virus, and then you can join this trial. And so we had enough students and lecturers and that coming down with colds to form you know, enough participants, and they went down to the laboratory, got themselves tested. Yes, you've got a virus. The average length of time that they already had symptoms was about 2.5 days.
Yeah.
And then they had to take either the placebo or the nucleotide supplement and report each day down to the laboratory to pick up their pills for the day, and also to take in their symptom scoring for the different things like sinus pain,
Yes,
runny nose, all the different symptoms of colds and flu.
yes.
And then so that they did that every day, and what they found was that those who had been taking the nucleotide supplement, there were 3 symptoms that had a significant impact reduction in terms of height of the severity of the symptom, and also the duration. And those symptoms were sinus pain, earache, and return to taste. So loss of taste. So for all those 3 symptoms, they reduced the severity, and also the speed of recovery was increased significantly. So we do know that taking, and it's a published trial, that taking nucleotides at an early stage of an infection, you can reduce the severity of your symptoms.
Very important. Yes.
Yeah, and we also know from studies that we've done, stress studies that we've done, intensive, moderate, and resistant stress studies with sports people, that we can reduce the build up of cortisol, blunt it, so there is less damage, but also what cortisol does is it depresses the immunity. So we managed to, what they tested for was the salivary IgA, and again we had a significant impact on preserving the salivary IgA during this extra stress. So there was less depression of the immune system in those trials.
One of our first lines of defense, isn't it?
Yeah.
Yes, of course. So that makes complete sense.
Yeah.
Absolutely. So
yeah.
Yes, so for those pesky opportunistic infections floating around, this could well be something really rather special and very well worth looking at. That is absolutely super, Rachel. Thank you very, very much. We'll leave it here for this module. And I just want to invite everybody back for our next module, module 5, which is where Rachel and I are going to take a dive into the other important roles of nucleotides. So as always, underneath this video, you'll see the little down arrow on the right-hand side. Just open that up, it'll give you the show notes. If you want to contact Rachel, if you have any questions, then please do so there. Or if you want to write some questions for us in the comments below, we can always get back to those as well. So thank you so much for being with us. Thank you, Rachel, once again, you've been amazing. And we look forward to seeing you all in module 5. See you soon.
See you later.